Good morning, everyone. My name is Diogo Gracia, and I'm a team leader of the scientific department of the VIII Bioengineering Week. First of all, I would like to thank you for attending our event today, and I hope you find it as interesting as we hope it to be. With that said, I present to you the role of microorganisms in a sustainable future lecture, which will be moderated by Professor Helena Pinheiro. Regarding the biocoins, do not forget to scan this QR code for a Google Forms and please fill out all the information so that you can earn the corresponding biocoins of this session and win one of the 3 prizes at the end of the week. If you cannot do it right now, do not worry because we can show the QR code again at the end of the session. Additionally, at the end of the 3 presentations, we will have a Q&A moment. So I invite you all to make some questions to our speakers, and these questions can be submitted to the corresponding section of the SBE app. Additionally, at the end, we will randomly select one of you to receive a small prize that we have prepared, so stay tuned. And with that, I hope that you enjoy this session, and I hand the word to Professor Helena.
I think I will stand for this also. So thank you very much. And I would first like to thank the organizing committee for inviting me to chair this session. I'm very honored, and I'm very glad to welcome our speakers and the audience, and to thank very much our speakers for being here and giving a little bit of their expertise to our audience. And of course, I'm very glad to chair a session with this theme, which I am sure that you are aware is one of the major themes for nowadays. So we are always thinking about sustainability. And in this case, we would like to make the future more sustainable. Of course, when we think about sustainability, we are already thinking about the future. So it's the essence of the word and of the concept. And in this case, we are looking into the role of microorganisms. And for that, we have different expertises here, from waste recycling, waste valorization, to the resources for macro and microalgae, and how the microorganisms are involved in letting us, in one hand, tackle those resources, so make them available for us. In another case, we would like to control them in order not to deprive us of our resources. And so this is quite a small group of speakers, but quite a wide, broad spectrum of applications of microorganisms, or the role of microorganisms in providing us with a sustainable future. So I will shut up now and do the introduction of our first speaker, who is Karina Felix, and she holds a BSc in Marine Biology and Biotechnology from Institute Polytechnic Leiria, and further MSc and PhD degrees in biology, with specializations in microbiology from the University of Aveiro. And she's presently a postdoctoral researcher as the Sete Mares, a very nice new building, Marine Sciences, Research and Development, Education and Knowledge Dissemination Center of the Mar Research Center of Institute Polytechnic Leiria at Peniche. And she's presently focusing on her research on bioactive compounds from seaweeds for several applications, particularly to control plant pathogens. So I give you the word, and thank you again very much, Karina, for being here.
So, first of all, good morning to you all. I would like to thank very much for the invitation. I'm really happy to be here. It's always a pleasure to share my experiences in this world of microbiology. So my name is Karina Felix, you already know at this point, and I'm going to talk a little bit of my experience and about what I'm doing now related with the role of microorganisms in a sustainable future. Okay, so the choices that I've been doing along the way. I really started at Polytechnic of Leiria in Peniche, and I did marine biology and biotechnology, and it was during this course that I found microbiology, and I fell in love. So I thought, okay, I must do this. And the next step was University of Aveiro, when I did my master and my PhD in the area of microbiology. So I started my master thesis with phytopathogenic fungi, and I decided to continue this subject for my PhD. So I had the opportunity to go deeper into that subject, which gave me a different perspective, I guess. Currently, I'm working at St. Mars in Peniche again at Polytechnic of Leiria, and I'm a researcher mixing a little bit of microbiology and marine biotechnology. And I will talk a little bit about this in the next slides. So at least for me, when I think about the role of microorganisms in a sustainable future, I go directly for bacteria and fungi and virus that can have a direct role. And I think that we will talk about that later. For example, in biocontrol, where we use microorganisms to control, for example, other pathogenic microorganisms. And then we'll have here a natural source controlling other natural sources. Another example is agriculture, where several microorganisms are known to be useful for soil fertilization or plant growth, because they can produce metabolites that are important, or they can degrade complex substrates to create availability to the plants and so on. The same is happening with waste management, where we can break organic matter into smaller compounds that can be useful for other organisms to use. And I actually added here the term biotechnology, but for me, all the other points can be also biotechnology, because they can occur naturally, but our mission as humans is to control everything. So we are exploring, we are going further, and we want to understand, okay, this microorganism can be useful for this or for that. And at this point, if we are manipulating something, we are talking about biotechnology. But here I added this point because there are several applications that are being explored nowadays. For example, for the production of enzymes, we know that, for example, fungi are very important in the production of several enzymes, because they can or degrade proteins or cellulose or something like that. And this is very important because several industries are already using these microorganisms to produce several enzymes. And when we talk about enzymes, we can talk about other things like pigments or other compounds. And finally, another very important term, that is bioremediation, that is so important nowadays, because this is when microorganisms have the ability to break down pollutants or pesticides or other bad compounds, helping to restore the affected ecosystems. So what I would like to do now is to look to a different perspective from my experience. And as I told you before, I started my master thesis, and I kept the same study for my PhD. And I was studying phytopathogenic fungi that is Lasiodiplodocea rumeae, typically known to infect plants. But with climate alterations, a few decades ago, they started to behave as human opportunists. So we wanted to understand how can this happen and why, and what may be the real impact of this. And for that, I used a really molecular approach using genomics, transcriptomics, metabolomics, proteomics, and so on. And due to that, I was able to deeper understand the behavior of these species. And for one side, I was able, when searching for all of these, the answer to these questions, I was able to find several enzymes that I was talking before that may have very interesting biotechnological potential. But at the same time, I was able to understand a little bit more the behavior of the species. And I think that this is also another perspective to look at how microorganisms can have a role in a sustainable future, because they are the base for everything, and we are several times creating conditions to turn, for example, a phytopathogenic fungus into a human opportunistic. So if we can understand the behavior of that species, we can predict some of the reactions that are coming, and we can avoid to use, for example, synthetic compounds or other type of strategies that are not so good for our planet. So I think that at this point it is very important to look further, to look deeper, and to understand the role of each microorganism, their mechanism of action, so we can use a different strategy to control them. So currently, as I said before, I am working with biotechnology, marine biotechnology, and also microbiology. And one of my main focus is on marine fungi. And this is in collaboration also with the University of Aveiro. And we are trying to find some marine fungi, to control phytopathogenic fungi. So fungi that are known to infect plants. And we already have actually good results in this. So maybe we can find or compounds or use the species, these marine species directly to do biocontrol. And this is a way to guarantee that we are not going for synthetic compounds and other prejudicial compounds for our planet. And also, I'm working at Polytechnic of Leiria with two different projects, and these projects are related with the exploitation of macroalgae and the bioactive compounds that these macroalgae have to find solutions, sustainable solutions, eco-friendly solutions to control. In the case of the project Orchestra, that is in the area of agriculture, we want to find bioactive compounds to control diseases that are associated to several orchards, mostly to maçã de Alcobaça and pera rocha, so you know it very well. And in the case of Cosmos, we are going to exploit a specific species of macroalgae, and the main goal is to find a solution, a sustainable solution for acne. So we can look at this and already see that we are trying to control microorganisms in a sustainable way, but we can also have the other side of the coin, that is macroalgae are not working alone. So we are extracting bioactive compounds from macroalgae, but they have also associated to them their own microbiota. So when we are extracting bioactive compounds from macroalgae, we must be aware that we are also extracting compounds that are produced from the synergy that come from microorganisms and macroalgae. So actually microorganisms are the basis for everything. I would like to finish my experience with a little bit different message, but that was really important for my experience. As we all know, a few years ago, we had this pandemic situation that was game-changing for everybody for 2 years, more or less. And I'm not an expertise in virus, but my team leaders, actually, in collaboration with other institutions, they were able to create a team. And in a record time, I would say, we found the solution for our West region. That was the creation of a diagnostic center for tests for COVID-19. And this was possible because, first of all, the knowledge about the virus that everyone that studied until there, and then the knowledge that we have in fundamental studies of microbiology and molecular biology and so on. So I think that I would like to share the message that we must look to the microorganisms as the basis for everything. We are probably doing, introducing some conditions in the equation that are changing the things. But if we look to them, if we study them, we will be able to understand them. And this is halfway to get and to find a sustainable future. So this is my contribution today. Thank you.
Thank you. Within the time. So we now switch from macroalgae, or at least part of macroalgae, to microalgae, and it's a very big pleasure to welcome Master Diana Gomes, who is a holder of a master degree in biological engineering here from Instituto Superior Técnico, and she is now working as a researcher in Alga for Future, A4F, which is a specialist, has been a specialist for quite a lot of years now in systems for the production of microalgae, and mostly in the development. So they are in the leading edge of developing microalgae production. And that's where Diana has been inserted in the team. And I'm very glad that she's here to tell us a little bit about her experience. So, Diana, thank you.
So, good morning, everyone. As you already know, my name is Diana Gomes, and I want to thank you for the invitation to be here today to talk a little bit about the role of microorganisms for a sustainable future. So first, I will start by talking about a little bit of my journey. So I did an integrated master's degree in biological engineering here at Instituto Superior Técnico. And in 2017, I did my master's degree dissertation in a partnership between Técnico and A4F, studying the extraction and characterization of microalgae proteins from an extremophile macroalgae. And I decided to study this topic because I already have a lot of curiosity to work with microalgae, so I decided that it would be a good team. And after I finished my master's degree dissertation, I joined A4F, and I'm working as a researcher at the Lisbon Innovation Laboratory, where I'm involved in several research and development projects. So about A4F. A4F is a company specialized in the process of design, build, operate, and transfer of commercial scale algae production facilities. And A4F is involved in several research and development projects, and it has a lot of experience in producing different microalgae strains. both at pilot and industrial scale. A4F is composed by the Lisbon Innovation Laboratory, which is a laboratory that supports some client and research and development projects. And we can do several activities, such as some analytical services. We can also develop some optimization of cultivation mediums. We can do microalgae strain isolation between other type of activities. And A4F also has the Lisbon Experimental Unit, which is a pilot unit that aims to mimic a microalgae production facility, where we have several different technologies for microalgae production. And we also give support for clients and for research and development activities. Then at A4F, we work with different types of photobioreactors. We can work with open systems, such as the raceways, and we also can work with closed systems, such as tubular photobioreactors and flat panels. A4F is also involved in the implementation of industrial-scale microalgae production facilities, such as the case of Alga Farm and Algatech Eco Business Park. So why should we think about working with microalgae and produce microalgae? Microalgae are photosynthetic microorganisms that only need sunlight, carbon dioxide, and water rich in nutrients in order for them to grow and produce oxygen. We have a lot of different species of microalgae. They can grow in freshwater environments and also in saltwater environments. And we have two types of microalgae, the prokaryotic microalgae that are called cyanobacteria, and the eukaryotic microalgae that we usually just call microalgae. So, the production of microalgae is very similar to agriculture because we only need water, sunlight, and carbon dioxide in order for them to grow. However, in the case of microalgae, we don't need arable land for their production. We don't compete with food crops production, and also we need a lot of water for microalgae production, but most of the water can be recycled. For example, when we harvest biomass, we get the biomass and then we can use the water that we recover to reintroduce again in the cycle of microalgae production. However, for microalgae production, we need a lot of carbon dioxide, and we also need some technology such as the photobioreactors and the harvesting equipment. Regarding microalgae composition, they are rich in proteins and they contain all the essential amino acids. They have polysaccharides such as starch and glucose. They have pigments like chlorophyll, carotenoids. They are rich in lipids such as polyunsaturated fatty acids. And they also contain all the essential vitamins. Due to this composition, microalgae can have several different applications, such as in the food and feed sector, in health and cosmetics, and also as biofertilizers. And there are also some emerging applications, such as for biofuels production, carbon dioxide mitigation, in wastewater treatment, and also in the production of biofibers and bioplastics. Now, I will show you two microalgae that you probably already heard about. They are the most famous microalgae. The first one is chlorella, that is a green microalgae, and you probably already see in some supermarkets, because this microalgae is very rich in protein, vitamins, and minerals, and is already approved as a food ingredient. And it also contains a lot of pigments like chlorophyll and beta-carotene that are important. Also, they can be very important. For example, if you ingest the microalgae, they can have some very antioxidant properties. For example, another microalgae that it's not a microalgae, it's a cyanobacteria. It's spirulina. Spirulina is very rich in protein. Almost 70% of its composition is protein, and is also approved as a food ingredient. And you probably also already saw in some supermarkets and incorporated in some other food. And spirulina is very known for the accumulation of phycocyanin. That is a pigment with a blue color, and this pigment can be used as, for example, a food coloring agent. And you have the example of the blue M&Ms that I don't know if you know, but they are the coloring agent is phycocyanin, and that's why it's blue. Now, I will talk a little bit about some projects that I've been involved and that show how the microalgae can be used in a more sustainable way. So the first project that I was involved when I joined A4F was the photofuel project. That is a research project which aims for the direct conversion of solar energy, carbon dioxide, and water for the production of biofuel precursors. So in this project, the objective was to cultivate 2 genetically modified microalgae. And these microalgae were modified in order to produce an alcohol that was butanol. And this alcohol was excreted to the culture medium. And then we can recover the alcohol from the culture medium and use it for biofuel as biofuel precursor. So the objective of A4F was to do the scale-up process of the genetically modified microalgae from laboratory to pilot scale, where we use 2 different types of photobioreactors, the flat panel and the tubular ones, and we were able to produce the biomass. But in this case, the objective was not to produce biomass, but to recover the alcohol from the to recover the alcohol from the cultivation medium. Another project that I'm currently working on is red wine. Red wine is a project which aims for the utilization of gaseous and liquid effluents from the wine fermentation and use it to produce chlorella biomass. And this biomass can then be used as food in the food, cosmetics, and agricultural sectors. So this project has an objective of a circular economy solution where we can use the wine fermentation effluents in order to produce microalgae, and then we can the wine producers can become also microalgae producers. So basically, the objective of A4F is to install in a winery at Palmela a chlorella production unit with different types of photobioreactors. And in this unit, we will use the gaseous effluent, which is rich in carbon dioxide, and also the liquid effluent, rich in several nutrients such as nitrogen and phosphorus. And these effluents will be incorporated in the production of chlorella. And the objective of this project is to produce, after the unit is optimized, is to produce at least 1 ton of biomass per year, to bioremediate 2 tons of carbon dioxide, and to treat almost 80 cubic meters of liquid effluent. So as I already told, this project will enable to valorize the effluents of the wine fermentation process, and with this, the wineries can also become microalgae producers and have 2 different types of products from their effluents. Then, an example of a partnership between A4F and a company that is ADP Fertilizantes. In this case, we will use microalgae for bioremediation. So at ADP Fertilizantes, they produce an effluent that is rich in nitrogen. And the objective of this project is to use the effluent rich in nitrogen as culture medium for the production of microalgae. And what will happen is that microalgae will incorporate the nutrients present in the effluent, and at the end, we will have a clean water and we will have a microalgae biomass. And then the biomass produced can be used as, for example, as a biofertilizer. Now, a more industrial project that we also have is Algatech Eco Business Park. This project is a partnership between 3 companies, Green Aqua Povo, which is the owner of the project. Then we have A4F that will give the technology and the production knowledge for microalgae production. And we have ICAM, that is a chemical company, former called Solvay Portugal, that will give infrastructures and also the utilities. So basically the objective of this project is to use the utilities produced by ICAM that are carbon dioxide, brine, water, and heat. And these utilities will be used for microalgae production using different types of photobioreactors. And at the end, we will have algae biomass that can be sold just as algae biomass, but it can also be used in a biorefinery in order to extract some algae-based products such as vegetable proteins, fatty acids, and antioxidant pigments. And this is an example how we can incorporate different industries in order to produce microalgae biomass that can then have several applications. To conclude, basically, we can use, if we have an industry that has some effluents that are rich in carbon dioxide or rich in nutrients such as nitrogen and phosphorus, we can use these effluents to produce microalgae biomass, which can have several different applications. And in the end, what we will have is we will reduce the industry's waste, and we will also valorize this industry's waste. And this is an example how we can enhance global sustainability. Thank you very much for your attention.
Okay, thank you. Thank you very much, Diana. And now we have our last speaker for this session, Anouk Duque, who holds an MSc in microbiology and a PhD in environmental science and engineering, both from Escola Superior de Biotecnologia da Universidade Católica no Porto. And she has worked in the wine and environmental protection industrial sectors, and has been an invited researcher at Delft in the Netherlands, at the Technical University of Delft. And she's presently a senior researcher at UCI Bio, Requinte, da Faculdade de Ciências e Tecnologia da Universidade Nova de Lisboa, working on waste biorefineries in collaboration with several industrial partners. They are very interested in that. So Anouk, thank you very much for coming. The floor is yours.
So, good morning, Paul. Thank you for the organizing committee for the invitation. I'm really happy to be here and to share with all of you a bit of my expertise and my research work I've been doing during the last years. Okay, so I could not find a better way to show you how much is needed to study microbiology. Okay, the planet is changing every day for a long time. So we need to find a way to fix it, because it's already there's not a way to come back. So we need to find processes to change and to get to a more sustainable planet. So the good news is that we live in a world full of microorganisms. And the amazing thing is that they can always adapt to the changes. Planet is changing, and they always find a way to adapt and to grow, which we can't. We can't. I don't know if you know, but you can find microorganisms everywhere, even in extreme environments. You cannot find human beings like this. So we should learn how to use the positive service of microorganisms for a sustainable future. But how can we do that? So I started my research path at Catholic University, as I'm a microbiologist, and then I did my PhD on wastewater treatment. So this is the first the first research team I worked, and starting from the beginning. I don't know if you know, but there is a lot of concerns about water. Water is scarce and is a valuable resource. And fortunately, the United Nations are thinking about it. So I don't know if you know, there is Agenda 2030, which states some sustainable development goals. So all the EU members have to meet the goals until 2030. And if you look there, we have water there's a lot of concerns around water that have to be solved. Okay, so microorganisms can help us in some way. Biological wastewater plants have a key role to return the water to the water cycle in a safe way. And why? Because they produce high quality water, because they use your microorganisms to remove the contaminants from waters. Okay? So it's an environmental friendly process and it's a sustainable process. When I started my PhD, I wanted to remove micropollutants from water. Okay, the micropollutants are pollutants that are in very low quantity, but they are very harmful, and some of them are even recalcitrant. But they are there, and then it's not anymore safe to return the water to the water cycle because they are still there, and then we need to remove them. So the first thing I did was to try to find a microorganism that was able to remove one specific micropollutant, which is 2,4-phenol. It's a compound that is present in pesticides. And after some months, I was able to do it because I went to a contaminated site, so in extreme conditions, to find soil samples that are rich in microorganisms that are able to live and grow in extreme conditions. So let's see if some microorganism here is able to degrade this compound. And we made it. And then we moved on. We tried to, we did a lab scale wastewater treatment plant, biological treatment plant. I chose rotating biological contactor, which is a series of discs with biofilm attached to the surface. And what we saw is that at the beginning, we could not have any biodegradation because we tried for around 100 days to put that microorganism in contact with the compound, and they were not able to do. They didn't die, but they were not able to remove this compound. Okay, so what we did is we add the bacteria we isolated to that system, and we found out that after adding the bacteria, 2,4-phenol started to be degraded. All the 2,4-phenol was degraded, so it was a good way to remove this compound, at least at lab scale. Okay, so this is called bioaugmentation. Then I went to Technical University of Delft, still doing my PhD, and I used, at that time, it was a novel technology. It was granular sludge technology. It's still a biofilm, but it's a special case. Microorganisms are able to grow attached to each other and form granules, as you can see in that picture. And I did the same. I tried at the beginning to see if these granules were able to degrade this micropollutant, and they were not. After more than 200 days in contact with it, even after this time, they were not able. So I also add the bacteria, and then all the compound was removed. The good news is, like with these results, we could see that the bacteria was able to attach to the granules. It's amazing. And then we could, in the real case scenario, we could have these kind of systems treating these micropollutants just by doing a bioaugmentation, adding the specific strain able to remove a specific compound. And you may be thinking, but why did you do the same kind of work with using biological contactors and granular sludge if biological contactors worked well? It's because this technology allows us to save space. When you use granular sludge, you can see here what you don't need anymore in your wastewater treatment plant. And this is only very important because you need to have a sustainable process in the way of the biological treatment, but also you need space. So it's a low footprint process. So we need to think in two ways. So you use the microorganism, and then the right microorganism to use the right process to also save space. And then since 2012, I'm at Nova University, and I'm working with plastics. It's a hot topic at the moment. Also, Agenda 2030 thought about that. You can see here plastic pollution in the oceans, not only in the oceans, but I think maybe you in the TV already saw lots of things about this, marine animals eating plastics and so on. So it has to be solved. You can see here, this exists, it's real. And also you have another goal that it's more to the solid waste, because you also have accumulation in landfills, lots of plastics. Okay. So any solution? We hope so. We hope so. So you can see that plastics are in your daily waste. If you look around, you have plastic everywhere. So they are essential. What is the problem? Because that they're petroleum-based compounds. They have a linear life cycle. So you use them, you dispose them. They are not degradable at all. It's an exhaustible source, because you may think, but petroleum is a renewable resource. It used to be, it's not anymore. It's a limited resource because you are using much faster than it's renewed. So it's not anymore a renewable resource. And you have the problem that is a recalcitrant material, which means that it's not biodegradable. It just stays in the nature. So here it comes polyhydroxyalkanoates. Polyhydroxyalkanoates are bio-based and biodegradable polymers. They have very similar characteristics to conventional plastics, and they are produced by microorganisms. So it's a green and a sustainable solution because they use microorganisms and they use ecological selection principles, and they are completely biodegradable and they are bio-based, so they are called the B2B plastics. So they are an amazing solution, I believe. This is a project where I was involved, one of the last projects that just finished a few time ago. So it's beer to biopol. So the idea was to valorize the brewery waste through polyhydroxyalkanoates production. This is a work in collaboration with several universities and with Superboc. They wanted us to use brewer spent grains, which is a byproduct of the process, very rich in cellulose and fiber, so it's very good to use for microorganisms. And they wanted to transform in PHA, polyhydroxyalkanoates, and then use it in their process, because I don't know if you know, they also sell water, so they need the bottles. So the idea was to use the byproduct of the beer production and then produce plastic bottles, PHA bottles, to use in the water. So they would close the cycle in their process. It's called the circular economy. And I'm just going to just show you an example. We are feeding the fermented beer spent grains I didn't put here, and you see the PHA, it's the green one, increasing. So bacteria were producing PHA. And then that picture just to see that how we can follow the process. We can do a staining with Nile blue, and then we see the cells shining under fluorescence lamp. And here you can have a few pictures of the PHA we developed in the end. So we are involved and are involved in several projects working for a more sustainable future. Some of them in bioplastics, some of them on wastewater treatment. Just you to know that lots of things are being done. And the take-home message that yes, microorganisms are very important. The key is to understand how they grow, how they manage to adapt to all these changes, and then we should use this in benefit for a more sustainable future. Okay. So, thank you.
Thank you again, Anouk, and thank you all for the insights that you have been bringing to us in your own areas of expertise. Very interesting, all of them. And we come back to the marine resources in the end, so that's nice. So how do we proceed? Okay, sorry. So we should start a discussion, and for that, some of you have already given start to it by posing some questions. And so what you have here as the first question is, what are the microalgae that you believe could be adopted for the food base for human consumption without being only regarded as feed supplements? So the basics, so like eating a steak or something like that, eating microalgae, instead of just being a supplement, like something to give you some antioxidant capacity or something like that. Diana, do you have any idea on that?
I think that one of the problems of just eating microalgae can be the flavor, because sometimes the flavor it's not very good. It's more like a fish flavor, and that's why it's easier to supplement things with a lot with low quantities, and it's easier for us to eat. But there are some research where we can extract compounds from the microalgae, and we can try, for example, some extracts have color, like green color, and when you use it to incorporate into food, then the food also gets that color, and sometimes it's not very good to look at, and you don't get you don't want to eat that because of the color. But there are some studies where they are trying to remove the color from the extract. And for example, you have a protein extract, and if you could remove the color and incorporate it into food, it becomes more pleasant to look, and you will have more. You want to eat it better than if it's green, and if it has that fishy smell, that's not so nice. But I think that one of the problems of just eating microalgae is that it's the smell and sometimes the color that is not so pleasant to use.
Not appealing.
Yes.
But do you have an idea if someone somewhere in the world is trying to develop some nice flavored algae?
There are some projects regarding that. I'm not I don't know a lot.
Okay.
But yes, there are some projects in that.
Okay, so there's hope.
Yes.
Okay, so for Karina, what was the result that surprised / amazed you the most during your PhD? So what was the wow factor in your PhD?
Actually, it was to find some molecules, some proteins and metabolites that were really specific for the infection of humans. And it was amazing. I was amazed by understanding that that species that I told you of that fungus in particular was able to produce those proteins or metabolites only when the growth temperature was higher, like 37 degrees, because it is our temperature. And when we expose the same fungus to 20-25 degrees, more or less the temperature for the plants, they were not able to express this kind of molecules. So this was the main finding for me.
Okay, yeah, thank you. Sorry, I haven't been telling you who is asking the question, but for Karina again, Mafalda Arnaud is asking, are these microorganisms in the microalgae cultivated in the lab, or do you have to study them based on their genome and some symbiosis with microalgae? So can you cultivate them? And what types of compounds arise from the result of their interaction that may be useful to acne?
We have several projects going on, and the project that is using more these particular bacteria and fungi that are in the surface of the microalgae are not any of the projects that I told you here. In fact, for those two projects, we are not extracting or cultivating the bacteria and the fungi, but it is possible to do it, for at least for some of them. We are actually treating the macroalgae as a whole, including everything. So I can tell you right now what are the compounds that are coming from the microbiome, or the compounds that are exclusively coming from the macroalgae. But we know that there are several specific compounds that are usually associated to the microbes that are there.
Still a lot to be
yeah.
researched. Thank you. So again, for Diana, do you think that it's possible to adapt the production of microalgae with an effluent rich in nitrogen to an industrial scale, which would be the bigger challenge?
Yeah, it's possible, and there are a lot of projects that try to do that. The bigger challenge, for example, in the project that I showed to you about the wine fermentation effluents, one of the biggest challenges is the seasonality. So you only produce a lot of effluent in a very small time. So you have, like, in the summer, you produce all the effluent, and in order to incorporate it in the microalgae production, you need to find a way to store that effluent, and it's a very big challenge because you can have changes in the effluent composition along the year. So it's an example of a big challenge regarding this type of use of microalgae. But if you have an effluent that is produced along the year, so it's easier to incorporate. And also you need to take into account that sometimes the composition of the effluent can be toxic for microalgae growth. So sometimes you need to dilute the effluent, and with that, you sometimes don't need a lot of effluent because you only need small amounts during the production. So I don't know.
That goes a little bit into another question that is asking how often do you need to replace the algae? So I'm not sure if I understand, but maybe replace meaning start with a new inocular, fresh inoculum. I don't know.
Well, it depends. You have a mode of production that is a semi-continuous mode. So you can do you periodically can harvest biomass, and you don't harvest everything. You leave a little bit of volume, and then you replace with culture medium, and then you continue the production. So it's a semi-continuous production. But how often you need to start again? It depends on the objective of your production. So if you have a lot of contaminants, for example, it's easier to start again and then to do this semi-continuous mode. But it always depends on the application that you want for the biomass.
Okay, so for Anouk, finally. So Rodrigo would like to know how does the process of harvesting and the modeling of the format of the PHA work? I'm not sure if I understand well, but please.
That depends
we can produce PhD. I don't know if it's the question, but we can produce using pure culture or mixed culture. I work with mixed culture, but part of my group works with pure culture. It's different ways of producing. Okay. In my case, I need a 3-stage process. I need 3 reactors. I need the first one when I use the feedstock. I use waste, so that's the advantage because I use a resource. I don't use synthetic. No, I even don't use novel substrates. I just go to industry and use their waste or my products. And I need to ferment it. So I need to transform it in organic acids, volatile fatty acids. And this is an anaerobic process. So we need to work to operate this first step in a way that we don't have methane. Because organic acid is just a step before production of methane. When I have these acids, I go to a second reactor, the sequencing batch reactor, aerobic reactor, where I could actually select for a culture able to produce PHA. I didn't tell you, but PHA, it's an amazing mechanism because microorganisms have this capacity to live in these extreme environments, as I told you. So if there's certain, I don't say all, but a certain kind of microorganisms that if they don't have the right conditions to grow, they just eat the carbon, but they store it in the form of PHA. When they have, again, conditions to grow, they will eat this PHA and they will grow. So it's they use it as a reserve for carbon and energy source. So, the scientists discovered that this PHA is that these granules, they are in granules inside bacteria, they have very similar properties to plastic. Okay, so during this second reactor, what we do is we have an amazing amount of microorganisms that we just go to a wastewater treatment plant, we get activated sludge and we put it in the reactor. And then we give it a selection mechanism to have, after a certain time, we have mostly bacteria or microorganisms able to produce this PHA. And when we have that, we have a third reactor where we actually produce PHA. So we let them produce until the maximum they can, and then we extract PHA. So this is the process that I use. Pure culture, it's much easier because you just have one microorganism, you just have one reactor, but you need sterile conditions, which is very expensive, and it's much difficult to upscale the process in that way because it will be very expensive. There are already also groups, part of my group also, I don't do that, but they are modeling the process, yes. But it's still running. It's not easy because all the microbiomes are different. Even though they produce the same, they don't behave the same. So it's work in progress. Work in progress. We still have to control some aspects of the process that are not being very so easy. And also, it's still an expensive process. The conventional plastics are around €1 per kilo. PHAs are still around €5 per kilo. So no one wants to go to the supermarket and buy something that would be much more expensive because of the packaging, even though it's sustainable. People are not, they're not educated for that. No one wants to spend more money. So we still need to work and to raise down the price of the plastic.
Yes, and I believe my personal opinion is that using mixed culture is the key to use wastes to bring the
I don't know if you noticed, even like the Kupal, they are changing
the straw.
To paper ones.
Yep.
But at the moment, it's the much easier solution is to go again to the paper. Of course, in the future, not so distant, it will be again another problem because paper was already a problem.
Yes.
It will be again, but this is a solution that they can take it now. Just go to substitute by paper, but it's not a sustainable solution.
Long term. Yes. It's just like a bridging solution.
Yeah.
Okay, Anouk, also, João would like to know if recently some news have been released about the presence of recalcitrant PFAS in water environments. And do you think it will be possible for microorganisms to remove them? And if there are any ongoing projects on that?
I believe, yes, I believe. I don't know if they are ongoing because now I'm not working at the moment with wastewater, but I believe so because there are always someone looking for micropollutants or even macro. But for sure it will be one. The problem is that I was lucky with my case. After 4 months, I had the microorganisms, but it will happen that it would not work. Just I was lucky. I went to the right side. I had the right sample, but so I believe that there will be always a microorganism able to degrade PFA or another pollutant, but we have to be lucky and have the right sample and the right enrichment and find it. But for sure, yes.
I believe that when we thought about using fluorine linking fluorine to compounds, to organic compounds, we created the most recalcitrant.
Yes. Good pesticides, bad.
Yes, but terrible now to undo what we have done.
Yes.
Sorry. One question that I forgot from Mafalda. How is the process of marketing algae as food ingredients? I'm not sure if A4F deals with that, or if they are thinking of creating a group that looks at it, at marketing of algae.
Yes, we have a company in our group that is Algae. That is a company that do the commercialization of the biomass that is produced at, for example, Algatech, and they are working a lot in the area of marketing the biomass, the microalgae, and selling it, and they are always looking for companies that want to buy the biomass. And that is the company in our group that is more focused in the marketing area. It's Algae.
Okay, yeah. From Leonor, do you believe that plastics degraded by microorganisms are close to a future solution? So I believe that Leonor means the actual non-biodegradable plastics. Can you actually find organisms like you found the ones for the fluorine compounds that can alleviate the problem of the plastics already in the environment?
I believe it's very difficult. This one is very difficult. They are really, even PET, I don't know if this is PET. So it's already something that is, part of it is bio. It takes 1,000 years to degrade. It degrades. So conventional ones, petroleum-based ones, I don't believe. There are lots of scientists, even this one, there are lots of scientists trying to get it faster, and they can't. So this one is, I don't think there is microorganisms able to degrade petroleum-based plastics, unfortunately.
Unfortunately. We really have to start at the other end, stop using the plastics and try to remove them, but physically, I think that
the problem is that we are dependent on the plastic.
Yes,
so it's difficult to move and to change. We don't have too much time because it's until 2030 that something has to be done. Also, supermarkets are working. So my group is working on that because they have until that they have to. They cannot have plastic. This kind of plastics in their shells, so they have to have biodegradable plastics. So they were also in a project with us but it's difficult because we can produce it, okay. And even we already have pilot plants working, and also we have partners in the industry that are also working, but the price is always a problem. Always. Because for the other part we have the solution. It's the price. Of course, this is being produced for years and years and years and years, so centuries. So this is an established process, completely optimized. That's why it's so cheap. These ones, it's a new process, so it will need some time to turn in a cheaper process. process. So, but converting biodegrading conventional plastics, I don't think so.
Yeah.
You just have to clean it.
Okay, for the end, João is asking when you have in bioreactors with aerobic or microorganisms, you need to transfer oxygen, and that's a major issue. So a possible solution is to use the algae to release the oxygen inside in the culture media. Do you apply this strategy? So do you combine the microorganisms that consume oxygen with the algae to provide that oxygen in your processes?
Yes, it's possible to do that, especially when we use the open system. We don't just have one microorganism in there. We have an ecosystem, and so in that case, we usually have that kind of process where microalgae produce the oxygen, and then other microorganisms use that oxygen for other processes.
Okay. Again, for Anouk, since the PHA accumulates intracellular, does it require cell rupture, or do the bacteria excrete it naturally? This is João, I think, asking.
It needs an extraction, so it's not so easy process because microorganisms are very smart. So you have to break the microorganisms, but you also have to break the granule, which is really well protected. Okay? So yes, it has to be extracted. We are also developing green, sustainable methods to do it because the conventional one is chloroform, so not really environmental friendly. And at pilot scale or even full scale, it uses hypochlorite, which is also not very friendly compound. So we just developed now a new extraction strategy with a bibliothetic solvent, very green one, and it worked very well. We already published, just published a paper on that, and we believe these people will move to this new strategy because it's also an important step of, actually it's the most important step in the process. Yes. Is to recover PHA, and it's also one of the most expensive steps is that the downstream processing. So we are also working on that. I believe we found a very good method to do it.
Yes. Thank you very much. I don't believe there are any new questions, but I have a question for Karine, and because we were talking about fungal pathogens, you were talking about that, and I was wondering because we have companies that are cultivating microalgae for food microalgae, to do the sushi and everything. And whether do you provide services? Are they also attacked by fungi, those cultures, or do you just use for land plants? So your expertise is more on the fungi that attack land plants.
Yeah.
Yes.
Well, they can attack, but I'm more focused, on land plants.
Yes.
So we are using macroalgae to basically extract the compounds because they are really rich in bioactive compounds with several different abilities, and they are being used in biotechnology for several industries. So we are using those bioactive compounds to try to fight against pathogens of land plants.
Eventually including aquatic plants. Yes. Okay.
Of course.
Yes.
If we find the right bioactive compounds, well, actually, in our projects, we have this concept to use a circular economy. So we have our main goal, but then if we find several bioactive compounds that they don't have the exact ability that we want, we will explore them to other activities. So yes, it's always a possibility.
because I'm thinking that actually using microalgae in as a fertilizer is an ancient practice. And they probably didn't know that they were doing that they were also transferring anti-fungi, so something that would benefit the health of their crops, isn't it? Yeah.
And nowadays we have several solutions that are already based in microalgae.
Mm-hmm.
We are trying to go further and extract bioactive compounds, because this way we can use not only an extract, we can be sure about that compound having that activity, and we can go further. But of course, that's something that has been used for a long, long time.
We're trying to do it again, but knowing a little bit more what
Yes.
we are doing. Yes, definitely. Well, thank you very much. And one thing that you have shown us is that you know what you're talking about. and thank you for giving us a little bit of that expertise and opening our vision on the possibility of using microorganisms or understanding the microorganism role. Thank you very much, and please. I give the floor back.
So we have reached the end of this interesting session. I want to thank once again our speakers for your willingness to be here with us today and share a little bit of your knowledge. Regarding our giveaway to today's lecture, we are giving an SBE mug, and the winner is Francisco Fragoeira. So please, you can at the end come here so that you can get your prize. And here is the QR code for those that have not scanned it yet. And I want to invite you all to our coffee break that we are having at the Congress Center. So thank you.
Eu acho que eu não conseguia achar toda a chance. Não sei se falhei alguma.